This chapter focuses on protein sorting into various organelles, and in this sub-lecture we're going to discuss the transport across membranes. So this, in our figure here of 3 different mechanisms of protein sorting, we're going to look at transport into the mitochondria, chloroplast, and the ER. So both mitochondria and chloroplasts have their own DNA and make some of their own proteins, but most of the proteins they get are imported from the cytosol. So they're made on ribosomes in the cytosol and then brought into the organelles by a process called translocation, where they're going through translocators in the membrane. So here's a translocator in the outer membrane, and then there's also one on the inner membrane. And they have to line up so that the protein can get through both the inner and the outer to the inside of the organelle. And as you can see, the protein gets stretched out or unfolded as it goes through the translocators. Because it does get unfolded, then when the protein reaches the inside of the organelle, there are chaperones, not shown in this diagram, but chaperones, other proteins that help the proteins refold. One other thing to note about this process is that the protein coming into the organelle has to have a signal sequence. That's a sequence on the end of the protein, and it's recognized by a receptor in the outer mitochondrial membrane. And it's a little hard for me to show you this to distinguish it, but this little piece of, let's see if I can make a circle around it. Little piece right here is the receptor for the signal sequence, and it's associated with the translocator, the protein translocator, that is going to transport the signal sequence across the outer mitochondrial membrane into the inner membrane space. And then this complex of the receptor, the protein, and the translocator diffuses in the membrane laterally in the outer membrane until the signal sequence is recognized by a second transporter or translocator, on the inner mitochondrial membrane. So saying they have to line up in order for the translocation to take place. Protein entry into the endoplasmic reticulum also involves protein translocators. This figure is just to remind us that there are these 2 different populations of ribosomes. There's the ribosomes that make proteins. The top level here, these ribosomes are located in the cytosol, and they're free ribosomes. And they make cytosolic proteins. And then there are the membrane-bound ribosomes that make proteins that are going to be inserted into the ER. So these proteins have a signal sequence in red here at the beginning that targets them to the ER, as they'll bind to receptors and to translocators in the membrane. So the signal sequence, that's the first part of the protein translated, directs all of these ER proteins to that organelle, and it's usually composed of 8 or more hydrophobic amino acids. As we'll see, it's going to be kind of inserted into the membrane. And then the protein is going to be kind of threaded across the membrane as it's being made. So kind of the energy to get it into that organelle comes from the synthesis. Kind of as it gets longer and longer, it pushes across. Now there are 2 protein components that guide the signal sequence. One is the signal recognition particle right here. That's in the cytosol. It binds to the ribosome and to the signal sequence. And then in the ER membrane, there's this signal recognition receptor, SRP receptor. The signal recognition particle receptor then associates with a protein translocator here in the membrane, and the ribosome, with the protein being synthesized, is transferred to this translocator, and the signal recognition particle and its receptor are released. Now, the signal sequence that was in the N-terminal of the protein, the red sequence at the end, is also important for holding the translocator channel open, so it remains there as the protein is first threaded through that translocator, and then it's removed by a peptidase, this yellow enzyme here, and it's released into the membrane. So here's that signal sequence, and gets eventually degraded. Meanwhile, as the protein is synthesized, it is threaded through the translocator. And this loop down here just gets bigger and bigger and bigger as the protein is made. And once synthesis is complete, then that translocated protein, it's released into the lumen of the ER, and the translocator closes, and the ribosome is released. And then again, there may be chaperones inside this lumen to help the protein fold into its mature conformation. Now, we know that membrane proteins can have some very complex topologies. They can cross the membrane one time, many times. They can be combinations of different monomers. There's a lot of different complexities to these proteins. But just to get an idea of how you can get, say, single pass versus multi-pass proteins, like they go across many times, I'll show you a couple of the control signals for this. So besides the signal sequence, the red sequence at the beginning of the protein, which is basically a start transfer sequence, that's what it's telling the translocator to do, is to open up when it binds to it and start transferring the newly synthesized protein through the translocator. So the translocator will continue to pass protein through, and as I said, this loop down here will get bigger and bigger until it reaches what's called a stop transfer sequence. So that's this yellow sequence in the protein. When it's made and meets the membrane or the translocator, the translocation stops, and then the translocator releases the growing protein. And as the ribosome can still continue to make the protein, but at this point it's no longer crossing the membrane. So the rest of the protein ends up on the cytosolic side. And so there, the process has created a protein that is not completely soluble in the lumen of the ER, but is a membrane protein that sits across the membrane. Now, in the case of a multi-pass membrane protein, such as this one going across the membrane twice, they have an internal signal sequence. So it's not right at the end of the growing protein, but partway into that protein, past the N-terminus. And this internal sequence acts as a start transfer signal, and it also anchors the final protein in the membrane. So it's not eventually cleaved like the signal sequences on the end of the protein. So this start transfer sequence, again, these are hydrophobic, inserts into the translocator and holds it open, and then protein is made, and the loop below the translocator grows until the stop transfer sequence is reached. Again, the translocator releases the protein. It's in the membrane, and it continues to be made until the C-terminal is reached. And so then the result is a membrane protein that passes over the membrane or through the membrane twice. And again, this signal sequence in red is not cleaved out. So in membrane proteins that pass through the membrane more than 2 times, there are just additional pairs of start and stop transfer sequences as the protein is being made. And they will come into play when they reach the membrane. A start transfer sequence will start the protein moving through the membrane again, and a stop transfer sequence will stop that transfer, and the protein will be made on the cytosolic side. So it can cross the membrane as many times as needed. It just has to have those start and stop transfer sequences. You might try to answer the question 15.4 that's in your textbook near these figures, because it will help you understand how these start and stop transfer sequences work, and also when you have how you can get an odd number of protein membrane spanning regions. Well, we saw how you could get 1, but how can you get 3 or 5? And so look at that question and see if you can figure out the answer.